All right, so this is Ed Latman speaking, and I'm just going to take a moment to introduce our speaker today, Dr. Roger Kornberg from Stanford University, although I don't know where he's actually speaking from at this moment. And Roger is professor of medicine in the Department of Structural Biology at Stanford. He did a BA at Harvard, a Ph.D. at Stanford. He was a postdoctoral fellow at the Laboratory of Molecular Biology in Cambridge. A wonderful history. And it was during the course of his stay in Cambridge that he made the original discovery of the nucleosome, the basic unit of DNA coiling in chromosomes. Roger's lab is involved in one of the most biological projects in our center. I think we have to admit that a lot of what we're doing is technology development and attempts to show proof and principle of certain kinds of projects. But the work that his laboratory is engaged in trying to get important structural information about the mediator complex represents, I think, the most adventurous biological project in our center, and it's been exciting for us to watch how it progresses. And I gather Roger is going to talk to us today in a more informal way about his history in science and how he views his role in the center. And thank you.
Thank you very much, Ed. I really appreciate it. And I was mentioning, Bill, at the outset that I would pause at some points in the course of what I say to invite questions so that there would be more of a feeling of connection with the online audience. Also, I understood in the spirit of these lectures to speak in a more historical way and tell you about the evolution of what we do and of my own scientific experience, because it is my understanding there are many young people who are listening. Perhaps the audience consists even primarily of students and fellows, and they may find it interesting to hear about the struggles as well as the successes. So I intend to convey that as well. And I thought I would go back to the very beginning, since you kindly recounted my history right from the outset, to when I was an undergraduate student, and mention what may seem surprising, that my undergraduate major subject was English literature. And I think it's worth a mention for the following reason, that the ability or practice and experience in communicating what we do in science is of such great importance. Very often I find that my junior colleagues work for years to accomplish something difficult. They solve challenging problems and then write up the work in a day or two or three or a week at most in a more or less complete but not really polished or, I would say, elegant manner. So they don't give nearly as much attention to the presentation of the work either in writing or also orally due to the work itself. And in that way, I think they fail to do themselves justice. And I've devoted enormous effort over the years to the writing up of what we do as well as its presentation. And I think one must not underestimate the importance. There's nothing wrong with spending months writing an important paper after you've spent years accomplishing the research. And at the end of the day, it makes all the difference, because whether or not anyone bothers to read and find out about what you've done depends on the way in which it is presented as well as the content of the material. I was actually reading a charming autobiography sent to me recently by Alan Heeger. Some of you will know Alan is a physicist now at UC Santa Barbara, I think in his 80s. Alan won the Nobel Prize in Chemistry, shared with two others, for the creation and development of electrically conducting polymers, organic molecules which conduct electricity as well or better than copper wires. And Alan comments that at the beginning of his career, wondering whether he should even pursue a career in science, he asked one of his professors, who was quite a senior figure, for advice, and the man asked him not about his background in science. He said, "How did you do in your English courses?" And Alan was taken aback, and then he explained, he said, the very thing that I've just told you now, that if you are not able to present what you do very well, don't bother to do it at all. Anyone, everyone, all of the young people who may be listening to this are capable of presenting very well. They just have to take the time and trouble and indeed engage in the struggle to do it. I mean, to give you an idea of how hard it is, when I started out, I could literally spend a full day trying to write one sentence and then the next day cross it out and start over. And it did take, as I have mentioned, literally months to write my first papers. So towards the end of my undergraduate experience studying English literature, I decided, I can't recall exactly why, not to continue, but to go to graduate school in chemistry. And at that point, I thought I would like, having studied mostly mathematics and physics alongside English as an undergraduate, I would look for someone who was especially skilled in that regard, but applying physical principle and knowledge of theory to chemistry, and especially as it relates to life science. And I looked around the country and identified who, in retrospect, was the ideal person for this, a man named Hardin McConnell, who those who have a background in chemistry, or especially more senior people listening, would know was truly one of the greatest chemists of the last 50 years. Hardin died recently in his 80s after publishing over 600 papers and making brilliant contributions, especially to the theory of magnetic resonance, both on nuclear and electron paramagnetic resonance. And thereby hangs, if you will, another lesson. I think it matters more who you choose to learn from than actually what you do. Hardin was really a theoretician. I mean, he did direct some experimental work, but he had no idea about the application to life science which I undertook. But it didn't really matter, even though he could never give me any specific advice about what to do. The period of years that I spent in proximity to him influenced me greatly, just being exposed to his way of thinking, the kinds of questions he asked, his critical approach to science. It was an enormous benefit. At the beginning of that time, he told me if I wanted to do something along the lines of what I mentioned, I would have to propose it, because it wasn't an area with which he was very familiar. So I decided, at that time, when some of you will be surprised to know, in the mid-1960s, the structure of cell membranes had not yet been solved, that I would investigate what was, I thought, the outstanding issue, namely how molecules cross the membrane barrier. And I thought, well, magnetic resonance is especially useful for measuring the rotation of molecules, including macromolecules. And perhaps I could discover how a membrane would bind a substrate on one side of a membrane, rotate, and discharge it on the opposite side. Well, it was in retrospect naive and impossible to do. And for that reason, I spent 3 fruitless years working not only harder than I ever did in the rest of my life, but harder than anyone has ever worked in my laboratory. 16-hour days, 365 days a year, really with great intensity. And at the end of 3 years, I had nothing to show for my efforts. which would probably alarm most young people. But I don't remember being discomfited by it, because I believed that eventually I would stumble on or discover something. And sure enough, one night during this long period that I mentioned, it occurred to me, why not simplify the problem? Instead of trying to discover the rotational motion of a large protein molecule of tens of thousands of mass units in a membrane, why not investigate the rotation of one of a lipid molecule? And this first slide will illustrate the point. So the first question I asked was, does a phospholipid of only about 1,000 molecular weight, so a few percent of the size of the proteins that are in the body, ever rotate? Does it ever, as I referred at the time, flip from one side to the other? I actually termed the process flip-flop because it related to a property of electronic circuits that I'd studied in physics as an undergraduate. Well, I won't go into detail regarding the measurement, which was made by paramagnetic resonance methods, but the conclusion was a lipid molecule almost never flips from one side of a membrane to the other. And I understood that this was because the polar head group, symbolized in this picture by blue spheres, is ionic and therefore insoluble in the hydrocarbon fluid interior of the bilayer membrane, which by about this time had been recognized as the organizing principle of all cell membranes. Hardin McConnell then said, well, if you've been able to estimate the rate, which was remarkably slow with a half-time of days, of passage across this short distance of a few tens of angstroms, why not measure the alternative process, which we refer to as lateral diffusion, namely the movement of lipid molecules within one leaflet, one side, one monolayer of the two that constitute the phospholipid bilayer of cell membranes. Using a nuclear resonance approach, I could determine that lateral diffusion happens about as rapidly as the movement of molecules in free solution. So at least 1 million, and we now know approximately 100 million times faster than diffusion across from one side of the membrane to the other. These measurements, which I made at that time, still stand as defining principles of lipid membrane or cell membrane dynamics. And I could go on in this vein, but rather what I'll tell you, at the end of that period, I thought, well, having learned a good deal about magnetic resonance methods, I'd like to become informed about the other important physical method for application of chemistry, and in particular physical chemistry, to life science, namely crystallography. This was now at the end of the 1960s, beginning of the 1970s, only a few years before the first structures of proteins had been solved by crystallography by Kendra and Perez in Cambridge. The Laboratory of Molecular Biology that Dr. Latman mentioned in the introduction was still method for the study and development of crystallography. And I applied to work with a man named Aaron Klug, who had done something innovative. He was a brilliant crystallographer, but also especially saw the opportunity of applying the approaches and principles of protein crystallography, of what had been developed by Kendra and Perutz, to scattering not of X-rays but of electrons. And so for the solution of structures by electron microscopy, which now, almost 50 years later, has finally, as many of you will know, come into its own as a really meaningful method for structure determination. I applied to Klug, and he accepted me to work in Cambridge. When I arrived there, I found out what they were studying by crystallography was especially the co-protein of tobacco mosaic virus, and then they were also at a late stage in pursuing the structure, the crystal structure of transfer RNA molecules. These projects were quite far advanced and not really very interesting from a biological standpoint, and I looked for something else to do. There was one person in the entire laboratory of molecular biology who was interested in biological membranes, their structure, their function, a man named Mark Brecher, not known to any of you, despite being a brilliant scientist, because he really devoted his entire career to one problem and worked primarily on his own and hasn't published much. But he had a great influence, and he said to me, well, why don't you read a paper that had been published by Francis Crick only a few months before. The title of the paper was A General Model for the Structure of Higher Organism Chromosomes. If you ever go back to that paper, and some of you may be amused to do so, it was published in Nature at the end of 1971, so a few months before my arrival in Cambridge in the spring of 1972. You will find, if you read that paper, that everything in the paper is wrong. It's written in Crick's magisterial style, and the logic is, as in everything he did, absolutely compelling. The paper is lucid, it makes fascinating reading, and everything about it is wrong. And it's an interesting question why it's wrong, and I could take time and explain it. It's really fascinating, but there's no time to do that now, only to say that the second figure in that paper shows a trivial idea for what is called the structure of nucleohistone. The figure refers to what had been a problem of about 75 years standing, unsolved, of the organization of a relatively simple nucleic acid protein complex, which constitutes almost all of the mass of almost all chromosomal material in all eukaryotic organisms. Nucleohistone consists of roughly equal weights of DNA and of a set of histone proteins, very small molecules, a little over 10,000 molecular weight, 4 principal histone molecules, which went by bizarre names at the time, but were renamed a few years later and are now called, as many of you will know, H for histone 2A, H2B, H3, and H4. The problem seemed, on the face of it, straightforward. I mean, just 4 small histones in DNA. I thought surely one could make some progress towards understanding the organization of that material, and that it would be, as Aaron Klug later explained to me, a suitable object for investigation by crystallography to learn the principles of X-ray diffraction. Klug pointed out to me it is shown by Hardin, Richards, and Wilkins, the Morris-Wilkins of DNA structure determination, and by a man named Vittorio Luzzati, working at Paris at the time, if I'm not mistaken, that nucleohistone gave rise to a simple diffraction pattern, a low-angle diffraction pattern with maxima at 110, 55, 37, and 27 angstroms. And these were believed to be orders of the primary maximum, the primary reflection at 110 angstroms. So 110, then 55 for half, 37 for a third, 27 for a quarter. And on that basis, Pardon, Richards, and Wilkins have proposed the structure of nucleohistone. They proposed a simple helix of DNA. So the DNA coiled in was termed a superhelix, with a periodicity of 110 angstroms. And it was thought that the histones coat the DNA and constrain it in that superhelix. There was a vast literature on the subject, including many studies of the histones themselves, their interactions with one another. It was a literal morass. It was completely undecipherable. The literature showed that every histone appeared to interact with every other one and with itself, forming oligomers, polymers of all possible sizes. There was really nothing rational or nothing that could be gleaned from such studies, so people had been unsuccessful in pursuing the organization of the presumed superhelix and the coating by histones of the DNA double helix in the superhelix of the chromosomal material, or chromatin. I began following, as one must do, the published procedures. I purified the histones as had been described by two groups, by a man named Johns in England and by James Bonner and his group at Caltech. And of course, I could repeat what had been observed. So to continue where I left off, I'll jump to the next slide. As I have already explained, the chromatin, which is the chromosomal material, is made up of roughly equal weights of histones and DNA, 5 histone proteins, but the principal ones, H2A, H2B, H3, and H4. I realized as I followed the published procedures, they were quite unlike what I had ever encountered in the past for the isolation of proteins from natural sources. Always, as everyone knows, when one purifies a protein, or in particular an enzyme, and wishes to conserve its activity, one takes care to try and avoid denaturation. But the methods that were used to isolate the histones were deliberately denaturing. In fact, to separate the histones, that was absolutely necessary. Now, the goal of that work at that time had been to isolate the individual polypeptides for sequence analysis, which was also in its infancy. The first protein sequence had been determined only about 15 years before by Fred Sanger, and the histones were attractive objects because of their small size, only a little over 10,000, and among the very first proteins sequenced after Sanger's work on insulin were histones, especially H4, which gave the remarkable result. H4 from a bovine source, from calf thymus, was identical in sequence, except at 2 residues, which were conserved, with H4 from plants, in this case from cow peas. It's an astonishing example of conservation, which only heightened the mystery and the interest in the histone proteins and what they do. At about this time, in the beginning of 1973, so a few months after I arrived in Cambridge, a paper was published in Biochemical Biophysical Research Communications, BBRC, a journal probably none of the people listening has even heard of it anymore, by 2 people from South Africa, showing what was in effect a failed attempt to separate the histones, except using mild methods. Instead of extracting the histones from chromatin with strong acid or with concentrated guanidinium hydrochloride denaturing the proteins, they isolated the histones they separated them from the DNA at high ionic strength, so with 2 molar sodium chloride, which of course is generally not denaturing. And then they were unable to separate the histones completely from one another. They could only resolve them into 2 pairs, H3 and H4, and the other pair, H2A and H2B. I was struck by the clean separation into those 2 groups because of what I have told you, the vast literature showing that every histone interacted with every other, including itself in an indiscriminate manner. Yet here instead were 2 preparations of soluble pairs of histones. And so I thought, as a way of progressing in research along these lines, I would pursue those histone pairs, and thought perhaps I would discover that there was a unique dimer, in this case of H3 and H4, that I might crystallize, then do the protein crystallography I'd come to learn, and in that way gain insight into the chromatin structure problem. I might add that the intensive work done over the years had led to the conclusion there could be no unique arrangement, because the best efforts made to measure the relative amounts of the histones showed a wide variation in their relative amounts from one source or one tissue within an organism to another. And indeed, it was well understood at this time, 1973, that there was not going to be discovered any unique repeating arrangement of these molecules. And yet, as I say, I was struck by this failed attempt at isolation, that there was a clean separation into 2 groups, and these were soluble protein fractions. I went on and discovered by both equilibrium ultracentrifugation and the newly introduced method of chemical cross-linking from George Stark at Stanford, that in fact H3 and H4 interact in a double dimer, an H3-H4 tetramer, and that H2A and H2B form a simple dimeric oligomer. And then I found they could be mixed together with DNA to recreate that X-ray diffraction pattern, the low-angle X-ray pattern from chromatin, which, as I mentioned, showed maxima at 110, 55, 37, 27 angstroms. So on this basis, I could believe that these oligomers were meaningful. And then the rest was straightforward, because if indeed all H3 and H4, when isolated, occur in this form, it suggested that pairs of histones, 2 H3s, 2 H4s, and then by inference H2A and H2B, must be the fundamental unit of chromatin structure. One can go back and calculate from what I told you that there are equal masses of protein and DNA. Well, the amount of the number of DNA base pairs corresponding to that weight of histone, 8 histones, 2 of each type, is about 200 DNA base pairs. And it was on this basis that I put forward the idea not of a superhelix of DNA, but of a particulate structure of chromatin based on an oligomer of 8 histone molecules. And I could calculate that they should be associated with about 200 base pairs of DNA. And then what is most important, we knew from the work of Perutz and Kendrew on, for example, hemoglobin by Perutz, similar to the H3-H4 tetramer, only an alpha-2 beta-2 dimer of globin subunits, the protein's structure is generally globular. Proteins form condensed units without large holes passing through them where DNA might possibly thread, and therefore the DNA must be wrapped around the outside of such a particle. And that led to the picture that I put forward in 1974. The proposal for the existence of the nucleosome and its organization is shown here. Many years later, that idea was validated by high-resolution crystallography, and the rest is well known to you. I wanted, at this point, to go on and pursue an independent career regarding the function of the nucleosome and of higher organism chromosomes in general. It was immediately apparent that wrapping DNA around a set of 8 histone molecules would interfere with many DNA transactions, especially the most important one in chromosomes, namely transcription, the readout of the genetic information. There was reason to expect, and there was evidence that gradually emerged, only upon unraveling DNA from a nucleosome did it become available for interaction with principal RNA polymerase for transcription of all genes that code for proteins in eukaryotes, what we call RNA polymerase II. I should say that the work that we and also Michael Grunstein and his colleagues did during the course of the 1980s established indeed wrapping a promoter. So the start site for transcription of a gene by polymerase II in a nucleosome does prevent the initiation of transcription. The nucleosome serves as a general gene repressor. That is the fundamental principle of eukaryotic gene regulation. Histones turn off all genes, and then only those that are turned on by specific positive regulatory mechanisms are eventually expressed, transcribed, appear in the relevant tissues. So we and others have been interested in those positive regulatory mechanisms in the several decades since that time. Now, for a period of, by now, several decades, we have pursued especially the polymerase II transcription machinery. Over the course of the 1980s, 3 research groups, that of Robert Rader and his colleagues, then at Washington University in St. Louis, the group of Ronald and Joan Conaway, who were then at the Oklahoma Medical Research Foundation, and my colleagues at Stanford discovered what is shown on this slide, namely the molecules required for initiation of transcription by RNA polymerase II at a promoter in a naked DNA molecule, presumed to be the template for transcription after histones are somehow removed from a promoter to allow transcription. We found, and Rader and the Conaways as well, there were 6 required components: the RNA polymerase, and a set of additional proteins, in some cases multi-subunit protein complexes called TF2 for transcription factor for RNA polymerase II, B, D, E, F, and H. The polymerase alone is capable of unwinding the DNA double helix and of RNA synthesis. But polymerase alone is not capable of recognizing a start site for transcription or initiating the process, and it is for those essential functions that the general factors are required. Now, the logical, and indeed still the only approach for understanding the requirement for so many molecules and the mechanism of the process in detail is, of course, structural analysis, including the overall architecture, the arrangement of components, and their detailed atomic or near-atomic organization. We began with RNA polymerase II, and the work on these lines was begun by 2 postdoctoral colleagues, Seth Darst and Aled Edwards, in the late 1980s and at the beginning of the 1990s, when this constellation of proteins that I show in this slide had begun to emerge and be established. We began pursuing the structure of the RNA polymerase, itself a large multi-protein complex made of a dozen protein subunits, 12 subunits, with a mass greater than 500,000 Daltons, about 10 times the size of any protein structure that had been solved at the time, and clearly beyond reach of crystallography, NMR, whatever methods were available for structure determination at the time. The only possible way of gaining insight regarding the structure of polymerase was by electron microscopy, which was being employed and had really been introduced or developed by Aaron Klug, who I mentioned, and his colleagues in Cambridge for the study of large assemblies of proteins, in their case virus particles, but also muscle fibers and other examples of large multi-protein complexes. And at the time, and the way in which Klug pursued the problem, structural information could best be gained by electron crystallography, that is to say, application of the procedures developed for X-ray studies to ordered arrays of protein molecules that could be viewed in the electron microscope. So virus particles with their highly ordered protein shells were one example, muscle fibers another, but there was no general approach for studying any other large multi-protein assembly in an ordered array or a crystalline form. I had the idea, I'm going to skip over this slide and come back to it. I had the idea of exploiting what I had learned about the lateral diffusion of lipid molecules in membranes to create such ordered arrays suitable for electron microscopy. The idea is illustrated here. It was to adsorb a protein of interest, in our case RNA polymerase II, to a lipid molecule suitably chosen, in this case indicated by the red triangles, for binding the protein of interest. The lipid molecule arrayed in a monolayer, which is readily spread, it forms spontaneously at the air-water interface. The proteins, once absorbed, would be constrained in 2 dimensions as required for electron microscopy to create a specimen thin enough for transmission of the electron microscope beam, but nevertheless free to diffuse because of the rapid lateral diffusion of lipid molecules that I mentioned, and we hoped to crystallize. Seth Darst and Al Edwards showed RNA polymerase II adsorbed to a lipid layer would crystallize, form large, well-ordered 2-dimensional arrays suitable for electron crystallography. It soon emerged that the size of the crystals and the extent of diffraction, so the degree of order of the crystals, was not sufficient for structure determination at a meaningful resolution until Al Edwards found the reason was 2 of the subunits, the 4th and 7th largest subunits, had a tendency to dissociate or were present in less than stoichiometric amounts with the others. So the protein preparation was, as we made it for these purposes, heterogeneous, a mixture of polymerase with and some without those 2 subunits. When Seth Darst and Al Edwards removed those subunits by deletion of the gene for subunit 4 in yeast, which does not impair the actual transcription mechanism, only the initiation of the process, then very large, exceedingly well-ordered 2D crystals could be formed suitable for structure determination. One crucial result of this work was then the discovery of a form of the polymerase suitable for high-resolution analysis. Another unexpected finding was illustrated here. Al Edwards noticed, and Seth as well, that even at the low concentration, microgram quantities per milliliter of polymerase used to form 2D crystals, there was a tendency to add additional layers and form thicker or indeed three-dimensional crystals by a process of epitaxial growth. And this could be exploited, as Al especially did, to grow large single crystals suitable for X-ray analysis. At this point, Dave Bushnell became involved and became, I would say, the one person who sustained the work throughout the time, from the beginning of growing those large single crystals to the solution of the structure and the work that has been done to the present day. There were difficulties. At the beginning, in the early 90s, the crystals, the large single crystals, failed to diffract. Al Edwards realized this was because they were prone to oxidation. Even when diffraction was obtained, it was only to relatively modest resolution. It turned out the problem was extensive crystal polymorphism, variation of the unit cell along one of the axes by a considerable extent, 10 angstroms or more. And finally, when that problem was overcome, standard heavy atom derivatives could not be obtained, and quite a long search, which eventually settled upon iridium and other derivatives, was finally successful. And so what this illustrates is how long it took from the beginning to the end of the process. Another point that I wanted to bring to the attention of especially the young people who may be listening, it took 10 years from my discovery as a graduate student of lateral diffusion and lipid layers to the formation of 2D protein crystals on lipid layers, which was really the starting point for the crystallography of RNA polymerase II. It took another 10 years before 3D crystals were obtained, another 10 years before high-resolution diffraction data were collected and phased, especially with heavy atom clusters, followed by individual heavy atom derivatives. And it was finally in the year 2000 that Dave Bushnell, Patrick Kramer, and I could report the structure of RNA polymerase II at 2.8 angstroms resolution. Now, what I will do in the remaining minutes is go back to what I told you, whoops, let me just back up, before, about the polymerase transcription machinery. I explained at the beginning polymerase is only one of the components of a very much larger apparatus with many more molecules involved. And the question naturally arises, why such complexity? Some of you may know that in bacteria there's only RNA polymerase, or at most an additional single polypeptide called sigma protein. But in all eukaryotes from yeast to man, there are these many additional molecules, the largest of which, TF2H, is larger than the polymerase itself, made of 11 subunits with 4 catalytic activities. Why such complexity? Well, it's even worse than that. We believed at the time that we identified this machinery along with the Rader and Conaway groups that it was at least complete, capable not only of initiation but also of a response to regulatory influences. But then the early 1990s, my colleagues, graduate student Kelleher and postdocs Flanagan, Kim, and Bjorklund discovered more was required for the stimulation of transcription by a so-called gene activator protein. They found that what was first identified as an activity in a crude extract from yeast and then later resolved to homogeneity as a 1 million Dalton 21 protein complex that we referred to as mediator absolutely required for a response to regulatory influences, including stimulation of transcription by an activator protein. Mediator, we found over the course of the 1990s, is conserved from yeast to humans and plants. The purpose of this slide is just to illustrate for every mediator subunit, the 21 of which are listed on the left, there is a counterpart in higher organisms, in flies, in human cells, also in plant cells. And mediator is not only essential for communicating between enhancers through activator proteins to polymerase and promoters, it is required for all transcription of all genes in eukaryotes. And as the bullet at the bottom summarizes, mediator receives and processes all regulatory information that impinges upon the transcription machinery, transduces a regulatory signal to polymerase and the general factors of a promoter to enable and to control the amount, the extent, the timing of transcription of all genes in all cells. So again, the question is how to understand the complexity. 25 subunits of the general factors. We now know 21 subunits of mediator, which assemble with polymerase, and a giant object the size of a subunit of a ribosome, 3 million Daltons, at every RNA polymerase II promoter prior to every round of transcription. Now with that, I will leap, as it were, 20 years to the recent solution of the problem at the level of polymerase and the general factors. And I'll come to it very quickly, and I'll skip over many details of the polymerase structure and only tell you during only the last 3 years, my colleagues here, especially Kenji Murakami, also near Kallisman, Hans Elmland, again Dave Bushnell, and others have succeeded in assembling polymerase and all the GTFs with promoter DNA in a stable initiation complex that could be isolated and analyzed with regard to structure and function. In this case, crystallography has not yet proved possible, but a cryo-electron microscope structure at between 6 and 11 angstroms, depending on what part of the molecule one refers to in the electron density map, has been derived, and it is shown here with superimposed upon the gray portion of the map at the bottom, the crystal structure of RNA polymerase II, superimposed upon some of the other parts, especially at the left, in green, in red, in dark blue, in pink, structures coming from crystallography of the smallest of the general factors, and you see a near perfect fit to the electron density of this so-called pre-initiation complex. In addition, one could see unmistakably the location of the promoter DNA, which is indicated by superimposition upon the electron density map of a DNA double helix in the middle, where the arrow indicates the direction of transcription, the direction downstream along the gene whose promoter is bound in the form shown here. Now, a remarkable finding of this work was what is shown in the next slide. Excuse me a moment, I'm just going to, sorry about my phone. A remarkable is the DNA is sharply bent at 2 points. And I could explain to you later why that may be, but now if we view down the axis of the DNA double helix, so from the point where the arrow is located on the right back towards the left, you see what is shown here. The green circle is formed by the a model of DNA that is superimposed on the electron density. So this is a view down the axis. And the notable feature, the DNA doesn't touch polymerase. This was a surprising finding. Promoter DNA does not come in contact with RNA polymerase II at any point in a pre-initiation complex for RNA polymerase II transcription. It is only associated with the general factors. And in retrospect, we can then understand what is the reason at one level for the existence of the general factors and their complexity. It is what is summarized here. Many of you will know that double-stranded DNA is comparatively rigid. It has a persistence length of about 500 angstroms, so almost 200 between 1 and 200, about 150 DNA base pairs. And therefore, it must follow a straight path. It cannot bend to interact as required for binding to polymerase. I could not take time to show you, but a bend through 90 degrees is necessary to seat the DNA in the polymerase cleft for transcription. So the general factors perform the role of recognizing the promoter, bringing it to the polymerase, positioning it above the active center cleft of the enzyme. Then one of the subunits of the general factors is responsible for melting the DNA to expose the template strand for transcription. When the DNA is in single-stranded form, it can bend. It is flexible. The DNA then can interact with the enzyme for transcription. Now, the last thing I'll do is go back to a previous slide. Whoops, excuse me. And comment on the mediator. So I will have told you then still only a part of the story. We know the structure of polymerase and some of the general factors at high resolution. We know the structure of the complex of polymerase with the general factors at an intermediate 6 to 11 angstroms level of resolution. The challenge, as was mentioned in the introduction, the outstanding challenge is now to solve the structure of mediator, to understand how it performs its important role as a molecular computer to process, as indicated at the bottom of this slide, regulatory information and deliver a signal to polymerase and the general factors at a promoter to control transcription. Turn it on, turn it off, if on, at what level, at what place, at what time in the life of an organism. The structure determination of this giant molecular machine, this molecular computer that processes multiple inputs of regulatory information, is an outstanding problem, and the one which was mentioned at the outset, we have especially pursued in the collaboration with the many wonderful colleagues on the BioXFEL project. The work on mediator is challenging because although Philip Robinson, a postdoctoral fellow here, succeeded in forming single crystals, the diffraction was limited to about 20 angstroms or so resolution. We think due to a significant degree of disorder in the crystals. As a next step, Phil and Dave Bushnell took nanocrystals of mediator for exposure on the XFEL beam. The initial efforts were not successful, but the work continues. We believe that we may know how to make mediator in a more suitable form for that analysis, and we remain greatly enthusiastic about the pursuit of the mediator problem with the use of nanocrystals and exploiting the advantages of XFEL and what have you, and that will be, as I have said, one of the chief objectives of our work towards the eventual complete solution of this problem in the years to come. So with that, I will stop. I've spoken for almost an hour, and I would welcome any questions or comments or whatever intervention may come from the listening audience.